Students, in this lecture we are going to talk about reproduction in algae. So as all lower plants or algae or other divisions of the algae, reproduction in the chara takes place by the various means like vegetative mean, is asexual mean, or the sexual type of the reproduction can be present in chara. So vegetative, if we talk about the first type of the reproduction method which is present in the chara, that is vegetative type of the reproduction. You all know what is vegetative reproduction. We have discussed it before, that if the reproduction takes place by parts of by body parts of any organism, and if a certain part of any organism detached from the parent part and develops into a new individual, so such type of the reproduction can be regarded as a vegetative type of reproduction. So in chara, vegetative reproduction can be takes place by the following means. The first method is by means of bulbils. Now what are bulbils? Bulbils, basically they are just like potato tubers. As you might have seen the tubers or the buds in the potatoes, and you might have seen that those buds in the potato, when they get detached from the parent body or from the parent plant, so they just develop into a new individual. So bulbils are the structures, they are just like potato tubers and they behave like the potato tubers. So this is very easy to understand if you imagine potato tubers in your mind. Bulbils are the spherical or the oval tube-like structures which develop on the rhizoids. They basically develop on the root-type structure of the chara. On the lower nodes of the main axis, the bulbils on detachment from the parents germinates into new thallus. They're just like the potato tubers. Bulbils are the outgrowths, you can see. They are the outgrowths and they develop on the rhizoidal part of the chara, and when those outgrowths get detached from the parent plant, they just develop into a new individual. Oh, okay. Second mode of vegetative reproduction is amylum stars. What are amylum stars? In the some species of the chara, on the lower nodes of the main axis, develop multicellular star-shaped arrangement of the cells. In the picture, in the lower picture, you can see the bulbils. Bulbils are the outgrowths like tubers, potato tubers, and in the diagram B, you can see amylum stars. Amylum stars, basically, this is a multicellular star-shaped aggregate of the cells. These cells are full of the amylum starch. Basically, these cells contain stored food material like starch, and hence they are called amylum stars because they contain a type of the starch which is called as the amylum starch. That is why this structure is called as amylum stars and it looks like star. The amylum star do detachment from the plants develop into new chara. So such like the bulbils, when the amylum starch get detached from the parent plant, it develop into a new thalli or into a new individual of the chara. Okay, the third type of the vegetative reproduction is amorphous bulbils. Amorphous bulbils are the group, many cells irregular in shape, which develop on the lower node main axis, like in the various species of the chara. The amorphous bulbils are perinating structures. When the main plant dies under unfavorable conditions, these bulbils survive and make chara plants on return of the favorable conditions. Now here you need to understand what are amorphous bulbils exactly are. Amorphous bulbils basically are the structures which are the perinating structures. And what are the perinating structures and what is meant by the perination? Perination basically is the time period or is the ability of any organism like plants, mostly the plants and the algae, perennation is the ability of the plants or the algae which make them uh, make their survival possible during unfavorable conditions like drought when there is no water, or like you can say in the harsh environmental conditions when the temperature is too high or too low. During the harsh environmental conditions, certain perinating structures are developed in case of the plants or algae. So these perinating structures make the algae able to survive during unfavorable environmental conditions. So amorphous bulbs basically are the perinating structures. They allow the algae or they allow the chara to survive during unfavorable environmental conditions. Now the fourth type of the vegetative reproduction is secondary protonema. Secondary protonema basically are the thread-like filamentous structure which develops from the primary protonema or the basal cells of the rhizoids. Basically, protonema develop from the basal cells of the rhizoids or rhizome. Protonema. What is the protonema? First you need to know what is protonema. Protonema is a thread-like structure that form the early stage of the development of the gametophyte in the life cycle of algae. In the life cycle of the algae, protonema is a thread-like or tube-like structure and which forms the early stage of the development of the gametophyte. Means you can say protonema is the early stage of life cycle of algae. Okay. The second type of the reproduction in the chara, which is advanced type of the reproduction, that reproduction is sexual reproduction. Sexual reproduction in chara is of highly advanced oogamous type of reproduction. You already know what is oogamous type of the reproduction, in which there are male and female sex organs present, and they fuse together and make zygote, and after the formation of the zygote, the further development of the zygote occur, embryo formed, and after successive cell division, a new plant or the new algae is formed. So sexual reproduction in the chara is highly advanced type. In this, the sex organs are macroscopic and complex in organization. Male sex organ, in case of the chara, they are called as the antheridium or globule. You need to know the name of the male sex organs in case of the chara or female sex organs. The male sex organs are called antheridium or globule, and the female sex organs are called as oogonium or nucleule. Most of the chara species are homothallic. What is meant by the homothallic? Homothallic are those species in which male and female sex organs, here male and male sex organs is written, but you have to correct it. You have to write it male and female sex organs. Okay, by mistake it has been written over here. So homothallic is a condition or is a type of thallus in which male and female sex organs are born on the same node or the same algae. Means a same plant, if we talk about the plant, so same plant or the same thallus of the algae contains both sex organs. So such type of the thallus or such type of the plant is called as homothallic. Here examples are also mentioned over here. Some species are heterothallic. What is meant by heterothallic? Hetero means different. So if we talk about the heterothallic, so male and female sex organs are born on different plants or on the different thalli of algae. The sex organs arise on the branches of the limited growth. So we have talked about what are the branches of the limited growth in the chara. Sex organs basically develop on those branches which are called as the branches of the limited growth or the primary laterals. The nocule above the globule. The nocule basically present above the globule. The development of the globule and the nocule means the male and the female sex organs takes place simultaneously, but species globule matures before the nocule. You need to know that the globule matures before the nocule. Means the male sex organs mature early as compared to female sex organ. In the diagram you can see the globule and the nocule, they are present on the lateral, on the side branches or the branches of the limited growth. So first we will talk about the male sex organ, which is the globule. The globule is the large spherical red or the yellow structure. So this is the general appearance of the globule. So next is the development and the structure of the globule. The early development of the globule and the nocule is similar. The peripheral cell of the lower node of the primary lateral mean the branches of the limited growth. The peripheral cells of the primary lateral or the branches of the limited growth, they divides periclinally. Periclinal division is horizontal division. Cells divide horizontally to form an inner cell. Firstly, the peripheral cells mean the outer boundary cells of the primary lateral, they divides horizontally or periclinally to form an inner cell. The outer cell function as enthedial initial. So when the cell division takes place, the outermost cell is called as enthedial initial. What is enthedial initial? The enthedial initial is the first cell of the male sex organ, which is called as enthedium, and that enthedial initial will go on further division and the male sex organ will develop. So we are talking about the development of the male sex organ, how the cells are divided for the formation of the enthedium or the globule. So the outer cell, basically the peripheral cells of the primary lateral, the outer cell function as the enthedial initial, and the lower cell again divides a periclinal division. The outer cell that will not divide first, it will act as enthedial initial, which will in future will make enthedium, and the lower cells again divides a periclinal division. Out of these three cells form the lowermost function as an internodal cell. The lowermost cell is called as an internodal cell. The middle cell forms a basal nodal cells, and the uppermost cells function as an enthedial initial. So after the division of the peripheral cells, three types of the cells are formed: lower node cells, internodal cells, or enthedial initial. The middle basal node cell divides to make the 5 peripheral cells, and out of these 5 peripheral cells, the upper one develops into oogonium. Two lateral ones form the unicellular bracteoles. Bracteoles basically are the small leaves-like structure which provides a protection to the gametophyte, and the two lower ones on either side of the oogonium forms cortex or remains non-functional. In the figure 8c, you can see this type of the division. Enthedial initial, and the lower cell will change into oogonial initial. Means the upper cell will make the enthedium or the male sex organ, and the lower cell is going to make the female sex organ or nocule or the oogonium, right? So in the diagram B, you can see the peripheral cells, they divide and make the enthedial initial, middle one the basal nodal cell, and the last one is internodal cell. Right. The enthedial initial divides by the transverse division. Now, after the periclinal division or the horizontal division, the enthedial initial cell, the in this diagram, the uppermost cell, it divides by the transverse division to make the basal pedicel cell and a terminal enthedial mother cell. Now these divisions are just for the formation of male sex organ. You know, every cell or the body of the every organism is made up by cells, and how these cells, how the body is formed after the successive cell division. So the enthedial initial divides by the transverse division. Transverse division are vertical divisions to make the basal cell and a terminal enthedial mother cell. In the figure 9a, you can see this apical cell, the uppermost cell, it divides periclinally and form enthedial initial and pedicel cell. There is a pedicel cell which is below the enthedial mother cell. The pedicel cell, the lower cell, the pedicel cell, it will not divide further and form a pedicel or stalk-like structure of the mature enthedium. The lower cell or the pedicel cell will not divide further and will make a stalk-like structure of the mature enthedium. The enthedial mother cell or the uppermost cell, it will divide and divides by the two successive vertical divisions at right angle to each other and make a quadrant. Okay, now the enthedial mother cell, it will further divide and form a quadrant. Quadrant means it will make 4 cells. All these 4 cells divide by the transverse wall to make 8 cells or the octant stage. Now those 4 cells will further divide and make octant stage or the 8 cells, each cells of the octant cells. Now those octant cells will further divide periclinally and forms the 2 layers of the 8 cells each. Okay. As in the diagram, you can see the 4 cells, the upper 4 cells will further divide, means the cell division is a continuous process. These 4 cells further divide, and in the figure F you can see those 4 cells further goes on division and make 8 cells which form an octet. And now in the octet, all the 8 cells will further divide and make in the figure G, you can see they make a filamentous type of structure, means cell division is a continuous process in the formation of male sex organ or enthedium. The cells of the inner or the outer layer divides periclinally to make the 3 radial layers of the 8 cells each. Now every cells divide and make 8 cells. Each cells will divide and division goes on until 8 cells are made from each cell. The outermost 8 cells enlarge laterally to form a curved plate of the 8 shield cells. Now the outermost layers of the cell, it enlarge and form a shield of cells. The cells of the middle layer elongate towards the center to make the 8 rod-shaped manobrial cells. The middle cells form rod-shaped structure which is called as manobrial cells. The cells of the inner layer function as 8 primary capitulum cells which will further going to divide into gametophyte. Each primary capitulum cells divide to form 6 secondary capitulum cells. Sometimes the secondary capitulum cells divide to make the tertiary capitulum cells. Here in the diagram H, you can see the complex type of the structure, and that complex type of the structure is called as primary, secondary and the tertiary capitulum. Each capitulum cells divide. Now cell division is a continue. Cell division each capitulum cell divides repeatedly to form the 2 to 4 long multicellular branched or the unbranched enthedial filaments. Now the capitulum is a structure which goes on the further division and it will form enthedial filaments or the spermatogenous filaments. In the figure G or H you can observe it. The enthedial filament has up to 250 uninucleate cells. Means the enthedial filament which are formed, they have up to 250 uninucleate cells, mean they have up to 250 cells and all cells have their own nucleus. These cells function as a sperm mother cells. All these cells function as the sperm mother cells, means in the future they are going to make sperm, and each cells give rise to a single spirally coiled uninucleate biflagellated entherozoid. Entherozoid basically is a cell which is called as a sperm mother cell. The mature globule thus is made up of 8 curved sheet cells. Now when the mature globule or the mature male reproductive structure is formed, it is made up of 8 curved sheet cells, 8 elongated manubrial cells, 8 centrally located primary capitulum cells and 48 secondary capitulum cells. So after a lot of cell divisions takes place, when a mature globule is formed, it has such amount of the cells which are mentioned over here. The secondary capitulum cells give rise to many enthedial filaments. Each sperm mother cell form a single by each sperm mother cell a single biflagellated entherozoid which will further form the enthedium is present. So this is a procedure for the development of the enthedium. So after the formation of the enthedium basically is a structure, is a male reproductive structure which contain the entherozoid. Entherozoid are the sperm-like structure which takes place in the fertilization. So how the entherozoids are liberated from the enthedium? Means enthedium is a cup-like structure, or that enthedium contain the entherozoids or the sperm. At maturity, the shield cells of the enthedium separate from each other. When enthedium is mature, the outermost shield cell, they separate from each other, exposing the enthedial filaments in water. The sperm mother cell gelatinizes to liberate the entherozoids. So this is the structure and the development of the male reproductive structure, which is enthedium. Now the second one is the female reproductive structure in the chara, which is nocule. The nocule of the chara is large green oval structure with short stalk. The stalk of the enthedium is large while the stalk of the nocule is short. It is born at the node of the primary lateral. It lies just above the globule. So its location is above the globule in homothelic species, means the species in which both male and the female reproductive organs are present. Now coming towards the development and the structure of the nocule. The upper peripheral, this is somewhat similar to the development and structure of the enthedium. So you will not have much difficulty to understand it. In this case, the upper peripheral cells of the basal node of the enthedium function as the oogonial initial. So now the peripheral cell of the basal node of the enthedium, it function as the oogonial initial, means the upper peripheral cell of the enthedium, it function as the first cell or the mother cell which will make a female reproductive structure like nocule or the oogonium. The oogonial initial, the first cell or the mother cell of the oogonium, it divides by the two transverse divisions to make the three-celled filament. In the diagram you can see the oogonial initial, it divides by the three transverse division and means a horizontal division and form a three-celled filament. You can see the three-celled filament in the diagram. It has a lower pedicel cell. The lower one is a pedicel cell just like the enthedium, the middle nodal cell and the upper oogonial mother cell. Now the basic function is of the oogonial mother cell, which will further convert into oogonium. The pedicel cell, just like the Enthridium, it will not divide further and make the pedicel or the stalk-like structure of the oogonium. The middle nodal cell under many vertical divisions to make the 5 sheath cells. Now the middle nodal cells, it will go under vertical divisions or form 5 sheath cells or the peripheral cell which surrounds the central cell. In the diagram D and F, you can observe the division of internodal cell. The central cell does not divide. The central cell will not divide and it will function as a node of the oogonium. The oogonial mother cell means the uppermost cell. It will further divide, elongates vertically and divides by the transverse division to make the lower small stalk cell and an upper large oogonium. So the oogonium contains a uninucleate ovum or egg. Now the oogonium, in the figure 10 G or H, you can see the oogonium. It contains egg or ovum. The peripheral cells or the 5 sheath initials elongates, means the surrounding cells which surrounds the egg cells, they are called as the peripheral cells or the 5 sheath cells. They elongate, they protect the egg cells, they elongate and divide by the transverse division to make tiers of the 5 cells each. The 5 upper tier cells form the coronary cells, which form the corona of the nucleus. In the diagram, you can see the corona of the nucleus or the coronary cells. Basically, they are the protective cells which protect the egg. The 5 lower tiers function as the tube cells. The lower tier cells, they function as the tube cells. The tube cells elongate and get spirally twisted in the clockwise direction on the oogonium. The nucleus migrates on the lower side and the receptive spot develops at the tip of the oogonium. Now the large amount of the starch and the oil get deposited in the oogonium because for the development of the oogonium, large amount of the storage material like starch and oil deposited in it. The mature nucleus is attached to the node by the pedicel cell. Pedicel basically is a stalk, the lower portion of the nucleus, and the mature nucleus is attached to the node by the pedicel cell. The nucleus is surrounded by the 5 tube cells. The tips of the tube cells form the corona at the top of the nucleus. The upper portion of the nucleus is called as the corona. The oogonial cell possesses a single large egg or the ovum, means they contain a single large egg which is going to be fertilized by the sperm cells or entherozoids. The nucleus contains a large amount of the starch and the oil. The receptive spot is present at the upper part of the nucleus. So this is all about the structure and the development of the enthridium and nucleus in case of chara. Okay, after the development of male and reproductive sex organs, fertilization takes place. You all know what is fertilization. Fertilization is a fusion of male and female sex organs to form the zygote, which will further develop into a new organism or a new plant or a new algae, any type of organism. So in fertilization, there is a fusion of the male and female gametes or male and female sex cells. After the development of the male and the female sex organs, the male and female sex cells in chara, they fuse together and they going to fertilize for the formation of zygote. So when the oogonium is mature, the 5 tube cells get separated from each other, forming the narrow slits between them. The 5 entherozoids are chemotactically attracted towards ovum. in case of the chara, how the ovum and the enterocytes, mean how the egg and the sperms, they are attracted to each other. The attraction is chemically. The attraction is going to done by chemical means. Certain sort of the chemicals basically are the reasons for their attraction. So the attraction of the sex cells, if it is done by means of any sort of the chemicals, so uh, such type of attraction is called as chemotactic attraction. The enterozoids enter through these slits. Enterozoids mean the sperms enter into the female reproductive organ by the slits and penetrate gelatinized wall of the oogonium. They penetrate into the jelly-like wall of the oogonium. The sperm cells or the enterozoids, they penetrate into the gelatinized wall of the oogonium. Many enterozoids enter oogonium, but only one enterozoid or only one sperm cells fertilize the egg to make a diploid zygote. There are number of the sperm cells, there are number of the enterozoids, they get entered into the oogonium or the female reproductive organ, but there is only one egg cell, one large egg cell, and only one sperm cell will get a chance to fertilize the egg to make a diploid zygote. Now the zygote secretes a thick wall around itself to make oospore. Oospore basically is a structure which is formed after the formation of the zygote, and that structure is a thick walled structure of the zygote. So coming towards the life cycle of the chara, if we talk about the general life cycle of the chara, so here is an overview of the life cycle of the chara. The plant body of the chara is haploid. The vegetative reproduction takes place by the formation of the vegetative mean of reproduction is done by amylum stars, bulbils and secondary protonema. Asexual reproduction is absent in chara. The sexual reproduction is advanced oogamous type. So this is the overview of the life of the reproduction type of the chara in general. The male and female sex organs are the globule and nucleus respectively. After fertilization, a diploid spore is formed, which is called as a zygote. At the time of the germination, diploid oospore, because it is formed by the fusion of the two gametes, so it is a diploid oospore. At the time of the germination, when a new plant is going to form, diploid oospore nucleus divides to make the haploid nucleate and the haploid chara plant. When the diploid oospore cell divides further, goes into the further meiotic divisions. When it goes into the further meiotic divisions, then it is ultimately formed a haploid chara plant because the meiosis formed the haploid cells. Thus, the life cycle of chara are predominantly haploid type. You can see because the plant of the chara is a haploid and its gamete is only diploid, so you can say that chara is a predominantly haploid type of the plant, or it has haploid type of the life cycle. In the diagram, you can observe all the structures we have discussed before in the structure and the development of the male and female sex organs. The main plant body is the haploid. It is mentioned by the single N. And in the branches of the limited growth, sex organs are developed, and these sex organs, when they develop, when they get mature, on the right side and on the left side, you can see the development of the nucleus and the globule. So when the male and the female sex organs, when they get mature, the egg and the sperm cells fertilized, and after the fertilization, what is formed, a diploid zygote is formed. In the diagram, you can see clearly, after the fertilization, a diploid zygote is formed, and that diploid zygote goes on reduction division, mean meiosis occur. Meiosis is a reduction division in which haploid cells are formed. Diploid zygote undergoes reduction division, which is called as a meiosis, and after the meiosis, 4 haploid cells or the 4 haploid nuclei are formed, and those 4 haploid nuclei the further develop into the lenticular cells. Means lenticular development is in which the apical cells, means the uppermost cells, are going to develop. After the lenticular cell development, rhizoidal and the protonemal initial development takes place, and after the rhizoidal and the protonemal initial development, a young rhizoid and the protonema, I have told you about it. Protonema basically is a thread-like structure which is the early stage of the development of the gametophyte. Means that protonema in the future will make a gametophyte, and after the protonema development, young rhizoid and the protonema is formed, and that young rhizoid and the protonema will going to develop into a plant body, which is a haploid plant of the chara. So this is all about the structure and the development of male and female sex organs or the sex cells in chara and its life cycle.